Okay, so the small intestines. The small intestines are referred to as your small bowel, and this is primarily due to the diameter. It's actually 6 meters long. It's much longer than the large intestines. It's the longest portion of the actual GI tract. And four main processes occur in the small intestine: secretion, digestion, absorption, and propulsion. I should mention in the stomach, and I forgot to mention this on the last recording, you're having mechanical digestion by the churning in the stomach. So you're continuing with the mechanical digestion. With the secretion of pepsin, you're also now starting to break down proteins, okay? Carbohydrates are also being broken down due to the hydrochloric acid. Lipids are not touched yet. Lipids will not be broken down or emulsified until they actually get into the small intestines, which we'll talk about in a few minutes. The cells of the small intestines are enterocytes, produce multiple digestive enzymes, hormones, and mucus. Enzymes, along with those released by the pancreas, are responsible for a bulk of the chemical digestion that actually occurs. No further nutrient digestion takes place by human enzymes once food leaves the small intestines. Note human enzymes, because once you get into the large intestines, you have bacteria, and bacteria can help to break down any food product that was not digested, not necessarily for absorption for you, but just to break it down. After nutrients are digested chemically, they are absorbed across the enterocytes into either the blood or the lymph. Fat is going to go in through the lymph. Proteins and carbohydrates will go in through the blood, along with water, vitamins, and other substances. The small intestines also mixes and propels its contents along the length and into the large intestines. Now, the small intestines has three distinct divisions. The duodenum is the initial segment and begins at the pylorus. It is the shortest of the three divisions at only about 25 centimeters or 10 inches long. It arches into a C-shaped as it curves around the pancreas, where only the proximal portion of the duodenum is within the peritoneum. The remainder sits on the posterior to the peritoneal cavity and is referred to as retroperitoneal. The duodenum will house the major duodenum papilla, which is where secretions from the gallbladder and the pancreas will enter into the small intestines. The gallbladder is going to be bringing bile, which will emulsify the fats, and the pancreas will bring in pancreatic juice, which will continue to break down proteins, carbohydrates, and also aid in the production of, or the breakdown of fats. The duodenal submucosa submucosa is going to contain specialized glands referred to as duodenal glands or Brunner's glands. These actually produce alkaline mucus to help protect the duodenum from the acidic chyme. Remember, the chyme leaving the stomach is going to be somewhere in the range of 2 for a pH. So, that can actually cause a lot of damage to the wall of the duodenum. So what these Brunner's glands do is they help to neutralize out that mucus, that secretion. The jejunum, and this is the middle segment, begins at the duodenal jejunal flexure, and it sits within the peritoneal cavity. It's measured at about 7.5 feet long in length. It is the most active site for chemical digestion and absorption. The ileum is the final segment of the small intestines. It is also intraperitoneal and measures about 10 feet long in length, up to 11 feet. It terminates at the portion of the large intestines referred to as a cecum, and you have what's referred to as the ileocecal valve. This is a sphincter that controls the movement of materials from the ileum to the cecum. This prevents materials in the large intestines from flowing backward into the ileum, and this is critical for preventing access to bacteria from the large intestine. So here we have our little C-shaped region here, the duodenum, the jejunum, and then the ileum here, and this will dump into the cecum. The internal surface of the small intestines contains 3 progressively smaller types of folds. Most absorption takes place in this region. The folds increase the available surface area for absorption about 400 to 600 times that of what you would normally think. The largest type of folds are visible with the naked eye, and they are referred to as circular folds, or the circularis. These involve both mucosa and submucosa of the small intestines, and they increase the surface area. They also slow down the transit of chyme through the small intestine, giving nutrients more time to be digested, and the small intestine cells, those enterocytes, more time to absorb those nutrients. And here you can see those circular folds right here. Okay. The smaller two types of folds are not visible with the naked eye. You have mucosa folds, which are into projections called villi. Each villus consists of a layer of enterocytes surrounded by a central core of blood capillaries and lymphatic vessels referred to as lacteoles. The lacteoles are where the fats will enter into the lymphatic system. Between the villi and the mucosa are indents to form intestinal crypts. These will house glands with both enteroendocrine cells as well as mucus-secreting goblet cells. And here you can see a villi on the surface. You can see the cells lining here. And along this entire single cell surface, you can see that you're going to have a capillary bed as well as a lymphatic capillary referred to as a lacteal. And essentially the nutrients would be absorbed through here. The smallest folds are referred to as microvilli, and these are actually in the actual plasma membrane. These are actual invaginations of the plasma membrane of the enterocytes. Each enterocyte can have as many as 3,000 microvilli, and this gives the cells the appearance of a bristle brush or brush border. Digestive enzymes are produced and secreted by the enterocytes, catalyze reactions that break down disaccharides and peptides. So again, all these folds, these three layers of folds, actually increase your overall surface area as high as 600-fold. The small intestine motility differs during fasting and eating. During fasting, small intestines exhibit slow, rhythmic contractions along its length in a pattern referred to as migrating motor complex. The contractions clear any remaining intestinal material, including leftover food and secretions. It requires about 2 hours to push digested food through the duodenum to the ileocecal valve. This is all controlled by both the enteric nervous system and motilin, which is a hormone that is produced in the cells in the duodenal mucosa. The small intestines will undergo two types of movement during eating. Vagus nerve appears to regulate both peristalsis and another process called segmentation, which I had mentioned earlier, I think in recording 1. Peristalsis is alternating contractions of longitudinal and circular layers of the smooth muscle in the muscularis externa. The primary function is to propel chyme towards the ileum and ultimately through the ileocecal valve to the cecum. Segmentation is actually intestinal churning. Contractions of only the circular layer of the smooth muscle. This produces a squeezing motion, and this primarily functions are mechanical digestion and mixing of chyme within the intestinal and the pancreatic enzymes as well as bile. So here you can see peristalsis, where it's a continuous wave-like motion, and here's more of segmentation, where you're sort of just having a circular muscle that's actually contracting there. This also is just sort of, I know we talked about several different types of hormones that are involved along the entire GI tract here, and this is just a summary of all of those hormones there. Now, appendicitis, you have the small size of the appendix and the fact that it is a blind-ended cause to cause it to occasionally become blocked, and it typically gets blocked by fecal material. Bacteria within feces multiply in the appendix, causing an infection. This results in inflammation, which is appendicitis. Signs and symptoms of appendicitis include abdominal pain, particularly over the right lower quadrant, rebound tenderness, similar to that experience with peritonitis, nausea, and vomiting. This requires immediate treatment, and most likely appendicitis actually requires, the treatment for this is removal. You want to remove this also before it bursts, because if the appendix bursts, you're now releasing boatloads of bacteria into sterile site areas, which could cause sepsis. If left untreated, this can rupture, causing internal bleeding, the spillover of the bacteria, fecal material into the peritoneal cavity, and this could be fatal if not properly managed and cleaned out. Okay, the large intestines. The large bowel runs along the border of the abdominal cavity, surrounding the small intestines and other abdominal organs. It measures about 5 feet long, and it's referred to as the large intestines again because of its larger diameter compared to the small intestines. This receives material from the small intestines that was not digested or absorbed, and it is actually a passageway for feces or fecal material to exit the body. Now, you're not actually going to absorb any nutrients by the large intestines, but the large intestines is actually involved in actively absorbing water and electrolytes, which is critical for maintaining fluid and electrolyte balance. It tests the large intestines to also include secretion, primarily in the form of mucus, propulsion, and defecation. It houses numerous bacteria that perform important functions such as synthesizing various types of vitamins. In fact, it's estimated that you have almost 10 times as many bacteria in your large intestines than you have human cells throughout your whole body. The different regions of the large intestines include the cecum, which is the first portion of the large intestines. It's a blind pouch located in the right lower quadrant of the abdominal area. It is also intraperitoneal. It's going to feature a small blind-ended pouch that extended from the posterior inferior end, which is referred to as the appendix. Generally shortened as the appendix, the vermiform appendix, which is generally shortened as the appendix. And again, the appendix will house multiple lymphatic nodules, and it plays a role in immune response. You then have the colon. This is the next and longest portion of the large intestine, and is divided into 4 portions. This is a retroperitoneal structure where you have the ascending colon travels superior along the right side of the abdomen from the right lower quadrant to the right upper quadrant. When it reaches the liver, it makes a sharp left-hand turn at the junction called a hepatic flexure. At the hepatic flexure, the ascending colon becomes the transverse colon because it passes transversely across superiorly abdominal cavity, the superior abdominal cavity. At the spleen, it takes a sharp turn inferiorly at the junction referred to as a splenic flexure, and this will then transition into the descending colon, which passes along the left side of the abdominal cavity. In the lower left quadrant, the descending colon becomes an S-shaped sigmoid colon, and then you'll go into the anorectal area. So the final segment of the small intestines are going to empty into here. This is going to be your cecum right here, and this is your appendix, this little tiny guy right here. All right, so then you have your ascending colon, the hepatic flexure, transverse colon, splenic flexure, descending colon right here, and then you have your S-shaped sigmoid colon right here, and then this is going to be the anorectal area. So after the sigmoid colon, the large intestines continues as the rectum. This portion of the large intestines runs anterior to the sacrum, and it is retroperitoneal. The walls of the rectum feature horizontal folds referred to as rectal valves. This is going to allow passage of gas without risking simultaneous passage of feces. The rectum ends at the anal canal, and this is the last portion of the large intestines. The anal canal will pass through the levator ani muscle and the floor of the pelvic cavity. The walls will feature longitudinal grooves referred to as anal columns. The terminal portions of the anal canal have two sphincters, an involuntary internal anal sphincter. This simply thickens circular layers of muscularis externa, and this is supplied by parasympathetic motor neurons. That is that is smooth muscle. That is an involuntary control. You then have your voluntary external anal sphincter, which is composed of skeletal muscles. This sphincter is voluntary and is supplied by the somatic motor neurons controlled by the cerebral cortex. However, the autonomic nervous system can take control of this if necessary and cause for relaxation of the external anal sphincter. And here's your anal rectal area. Looking at the histology of the large intestines, there's going to be a little bit of a difference. The mucosa of the large intestines lacks villi, and the cells will actually lack microvilli. Structural adaptations will reflect the fact that nutrient absorption is not part of the large intestine's primary function. The mucosa is rich with goblet cells that secrete protective and lubricating mucus. What's happening here is you are removing a lot of fluid in what will become feces, and that becomes essentially a hardened structure. So the mucus will help to move the feces along the large intestines. The muscularis externa of the large intestines is unique in that its longitudinal layer is not continuous throughout the most of its length. This layer is gathered into three bands or ribbons of muscle referred to as tinae coli. Their constant tension bunches the colon into pockets referred to as haustra. Sometimes these pockets get too defined, and some people have very defined pockets then where things like seeds and nuts can get stuck, and that could actually cause for a lot of discomfort. The serosa of the large intestines contains fat-filled pouches referred to as the epiploic appendage. The large intestine also houses a staggering number of bacteria. Indeed, there are about 10 times more bacteria in the large intestines than cells in the human body. And feces is actually made up of 65% or 60% of the dry weight of feces. These bacteria, which are referred to as your microbiome or normal flora or gut flora, consist of about 500 different types of bacterial species that coexist with humans in a symbiotic, mutually beneficial relationship. Humans will provide bacteria with the environment that they need to survive. The bacteria perform a number of useful functions for humans, including production of vitamins and metabolizing any undigested materials. To deter the growth of harmful bacteria, the normal flora prevent the growth of pathogenic disease-causing microorganisms by competing for those nutrients. And there's actually a disease referred to as Clostridium difficile or C. diff. And this is when you have a very pathogenic bacteria that sort of has invaded that large intestines, and it causes a lot of disrupt for the patients and pretty nasty diarrhea. In problematic treatment cases, what they will actually do is what they call a fecal transplant, where they take fecal material from a healthy donor and transplant that in. And what happens is you're essentially putting trillions of good bacteria in, which will outcompete for that bad bacteria. Bacteria in the large intestines will also stimulate the immune system. During infancy, normal flora will induce immune tolerance to their own antigens. At the same time, they stimulate the development of mucosa-associated lymphoid tissue, MALT, and the production of antibodies to pathogens. The large intestines has two functional segments: the proximal large intestines, which consists of the ascending and transverse colon, and the distal large intestines, which consists of the descending and sigmoid colon, rectal, and anal canal. The proximal large intestines is primary site of water and electrolyte absorption and bacterial activity, and this exhibits two types of motility. The first is segmentation, which is that churning action. The circular muscles of each haustrum contracts repeatedly, swirls the material around in the haustrum. This aids in water and electrolyte absorption, primarily controlled by local neurons of the entero nervous system, which is triggered by stretch. The other type of motility is in the proximal large intestines, is propulsive motion known as mass movement or mass peristalsis. During mass movement, multiple haustra undergo peristalsis. This propels their contents towards the distal end of the large intestines. Mass movement occurs 3 to 4 times per day, and it appears to be triggered by food consumption and initiates by reflexes within the entero nervous system. The distal large intestines performs a small amount of absorption, primarily that of water, but its main role is to store fecal material until it can be expelled through the process of defecation. The distal large intestines is much less motile than the proximal large intestines. When mass movement forces fecal material into normally empty rectum, it then initiates parasympathetic mediated defecation reflex. The stretch receptors transmit sensation of rectal distension to the spinal cord. That will then have parasympathetic neurons causing smooth muscle in the sigmoid colon and the rectum to contract, and internal anal sphincter to relax. Impulses from the cerebral cortex will then trigger the relaxation of the external anal sphincter and the contraction of the levator ana muscles, in which then feces is expelled from the body. Anything that changes the motility of the large intestines affects the amount of water present in feces. When motility increases, large intestines does not have enough time to reabsorb the water from the fecal material, and it produces very watery feces referred to as diarrhea. Factors that may increase motility would be any irritation to the colon due to bacterial or viral infections, and drugs that stimulate parasympathetic nervous stimulation. However, when motility decreases, it absorbs too much water and the fecal material sort of becomes too hard and becomes lodged, and that is referred to as constipation. The large intestine's motility may be slowed by drugs such as an opiate, narcotics, or those that block the effects of acetylcholine. That's why a lot of people that are on pain medications sometimes also need to go on like Colace or something to keep things moving along. So this just goes over the various structures and functions of the organs of the GI tract. This is a great slide because it gives you the properties of each of those areas of the oral cavity, pharynx, esophagus, stomach, small, large intestines. And it also gives you their structural, their functional roles. So pretty much the last 150 slides can sort of be summarized up on this single slide here. I wouldn't just study the single slide though. All right, and this can be it for recording whatever this is. I think it's 3.